Welcome to today's lesson on osmosis, where we will explore the process of how water moves in and out of cells via a partially permeable membrane. In our lesson today, we will first define osmosis and discuss the concepts of dilute and concentrated solutions. We will also explain what a partially permeable membrane is and how it enables osmosis. Next, we will examine how the movement of water can be modelled using a partially permeable membrane bag and sugar solution. Finally, we will discuss how different concentrations of solutions can affect water movement in and out of both animal and plant cells. So far, you have learned about the structure and function of cells, including the basic components and their roles. This foundational knowledge sets the stage for us to introduce the concept of osmosis, which is crucial for maintaining cell-water balance for regular function. We've also covered the role of diffusion, which is the movement of particles from an area of high concentration to an area of low concentration. We will now learn about a special case of diffusion, specifically the diffusion of water, which is called osmosis. Osmosis helps us understand how cells maintain their internal environment, which is essential for their survival. It also helps us comprehend various physiological processes in plants and animals, such as nutrient absorption and waste removal. To begin with, let's define what osmosis is. Osmosis is a diffusion of water across a partially permeable membrane. Specifically, it can be described as the movement of water molecules from a dilute solution to a concentrated solution through a partially permeable membrane. For osmosis to make sense, it's important to understand what we mean by dilute and concentrated solutions. In a dilute solution, there is a high concentration of water molecules and a low concentration of solute, like sugar. In contrast, a concentrated solution has a low concentration of water molecules and a high concentration of solute. A concentrated solution can become a diluted solution when additional water is added, as this results in a low concentration of solute in relation to water. We also need to understand what is meant by the term partially permeable membrane. A partially permeable membrane, such as a cell membrane, plays a crucial role in osmosis. This type of membrane allows only certain types of molecules to pass through, while blocking others. As we have seen before, the cell membrane in animal cells is partially permeable in that it allows certain products, such as glucose, water, and oxygen, into the cell, but prevents other, larger molecules from entering. In the case of osmosis, the partially permeable membrane allows water molecules to pass through, but prevents larger molecules like sugar from crossing. Another key point is that osmosis is a passive process. This means it does not require energy to occur. The movement of water is driven purely by the concentration gradient, moving from an area of high water concentration to an area of lower water concentration until equilibrium is reached. To better understand osmosis, let's consider how we can model it using a partially permeable membrane bag and sugar solution. By using a beaker of sugar solution, a glass tube, and a bag with a partially permeable membrane, we can model the process of osmosis in a cell. In this model, the water molecules can cross the partially permeable membrane, but the sugar molecules cannot. This means that water can move into or out of the bag, but sugar cannot. The movement of water through osmosis in this model will vary depending on the type of solutions that we use. In our first scenario, let's consider an isotonic solution. An isotonic solution is the term used to describe a solution which has the same concentration inside and outside of the bag. When this happens, there is no net movement of water, as the concentration is the same. Next, let's look at a hypertonic solution. If the concentration of sugar is higher outside the bag than inside, water will move out of the bag, and the final water level will be lower than where it began. Here, the outside solution is hypertonic to the inside of the bag, meaning it is more concentrated than inside the bag. Conversely, if the concentration of sugar is lower outside the bag than inside, water will move into the bag. The outside solution is then hypertonic to the contents of the bag, meaning it is more dilute than inside the bag. In this circumstance, water moves into the bag so that the final level of water is higher than where it began. So, we have looked at what the process of osmosis entails. However, we need to be aware of its application and significance in biological processes. In animal cells, osmosis has significant implications due to the absence of a rigid cell wall. Let's take a look at what happens in different situations where osmosis occurs. When an animal cell is placed in an isotonic solution, the concentration of the solution inside the cell is the same as the solution outside. Therefore, the cell retains its normal shape and functions as usual. When an animal cell is placed in a hypertonic solution, where the solute concentration outside the cell is higher than inside, water moves out of the cell. This causes the cell to lose water and shrivel. If too much water is lost, the cell can die from dehydration. In a hypertonic solution, where the solute concentration outside the cell is lower than inside, water moves into the cell. This influx of water causes the cell to swell and potentially burst because the cell membrane cannot withstand the excessive internal pressure. In light of this, we can see the importance of osmoregulation, which is the process by which cells balance the uptake and loss of water. The outcomes that can occur due to hypertonic or hypotonic solutions can be very dangerous for cell functioning. Osmosis also impacts plant cells. Plant cells, unlike animal cells, have a tough cell wall that provides additional support and stability. Osmosis plays a different but equally critical role here, with different potential impacts. When a plant cell is in a hypotonic solution, where the solute concentration outside the cell is lower than inside, water moves into the cell and fills the central vacuole, causing it to swell. This increase in water causes turgor pressure, which pushes the cell membrane against the cell wall. This turgor pressure is crucial for maintaining the rigidity of the plant, keeping it upright and firm. In an isotonic solution, where the concentration inside and outside the cell is equal, there is no net movement of water. The plant cell remains in a stable but non-turgid state. When placed in a hypertonic solution, where the solute concentration outside the cell is higher than inside, water leaves the plant cell. The cell becomes flaccid, meaning it loses its firmness and can wilt. If water continues to leave the plant cell, the cell membrane can move away from the cell wall in a process known as plasmolysis. This can lead to the death of the cell if the condition persists. Understanding osmosis is therefore essential because it affects how cells interact with their environment. For animals, improper osmosis can lead to cell damage or death, while for plants, it can affect their structure and ability to stand upright due to turgor pressure. Organisms must balance water intake to survive, and this is achieved through processes like osmoregulation in animals and the maintenance of turgor pressure in plants. Let's work through a question to apply our understanding of osmosis. If a partially permeable membrane bag contains a concentrated sugar solution and is placed in pure water, what will happen to the water movement? Pause here to have a go. Now, let's go through the answer. First, we need to identify the concentration gradient. The sugar solution inside the bag is more concentrated than the pure water outside. This means that the solution outside is hypertonic to the bag. Now we need to predict the movement of the water. Water will move into the bag from a region of higher water concentration, in this case pure water, to a region of lower water concentration, in this case sugar solution. Finally, we can determine what will happen. The bag will swell as it gains water. Before we wrap up, let's explore the historical study of osmosis. The study of osmosis took a significant leap forward with the pioneering work of German scientist Wilhelm Pfeffer in the late 19th century. Let's find out a bit more.
In the late 19th century, I developed one of the earliest devices to measure osmotic pressure, which later became known as the Pfeffer cell. I demonstrated the critical role of osmosis in maintaining the shape and function of plant cells. This research was not just about understanding plant physiology, it laid the foundation for how we perceive the interaction of cells with their environment and the maintenance of homeostasis. My findings opened doors for exploring osmosis in animal cells, shedding light on its vital role in cellular processes.
In conclusion, osmosis is the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. Solutions can be classified as isotonic, hypertonic, or hypertonic relative to the cell. all of which result in different water movements during osmosis. This passive process does not require energy and is essential for maintaining cellular function in both animal and plant cells. In animal cells, osmosis helps maintain cell shape and function, preventing cells from bursting or shriveling. In plant cells, it ensures cells remain turgid and the plant stays upright. Understanding osmosis allows us to appreciate the delicate balance needed to maintain healthy cells and, by extension, healthy organisms. By understanding these fundamental principles, you can gain insights into how osmosis affects the water balance of the cell. Using model systems, such as a partially permeable bag in solution, can help us understand how this process works. Watch the rest of the GCSE biology course at Sophos Education.